Hi, my name is Daniela Dian, and today I'm excited to be presenting to you my research on gene expression and heart regeneration. First off, why study heart attacks? In the US, 805,000 people have a heart attack each year. This means someone has a heart attack every 40 seconds. Now, a heart attack isn't just like scraping your knee or breaking a bone. Hearts do not have the ability to regenerate. When a heart attack occurs, there's a lack of blood flow to the heart, so it cannot adequately pump oxygen throughout the body, and there's scarring and a loss of function. Now, hearts do not have the ability to regenerate in humans, but other animals, such as zebrafish and amphibians, do have regenerative capacity and maintain this throughout their adult life. Now, what could be the reason for this? Is it that the hypoxic conditions they live in, that they're cold-blooded, that they just have lower order hearts? Well, interestingly enough, these animals have more mononuclear cardiomyocytes. The Patterson paper looked at the hybrid mouse diversity panel and saw a huge variability in the mononuclear cardiomyocyte percentage in different strains of mice. Here we can see on the x-axis 120 different strains of mice, and the percentage of mononuclear cardiomyocytes ranges from about 2% to 16%. Now, how does this variability affect heart regeneration? Well, if you look here at figure F, the paper chose two strains of mice with low mononuclear percentage, as indicated with the blue-purple lines, and two with high mononuclear cardiomyocyte percentage, the pink and red lines. A myocardial infarction was induced in all these mice strains. And as you can imagine, the ejection fraction, basically how much the heart is pumping blood, how well the heart is working, it decreased over the first 3 days. Now, in the mice strains with low mononuclear cardiomyocyte percentage, this decrease continued throughout the next month. In comparison, those with high mononuclear cardiomyocyte percentage had an increase in their ejection fraction, showing heart regeneration. This paper also went on to look at what gene could possibly be influencing mononuclear cardiomyocyte percentage, and they found TNNI3K. TNNI3K is a poorly understood heart-specific kinase. And so the Patterson paper created a TNNI3K knockout model. As you can see in the Western blot here, in the flux area, there's no TNNI3K present. Then they looked at how this affects the percent of mononuclear cardiomyocytes. And interestingly enough, this is an inverse relationship. The knockout of TNNI3K leads to increased mononuclear cardiomyocyte percentage, whereas in the wild type, when TNNI3K is expressed as normal, there is lower percent of mononuclear cardiomyocyte. This led to the question, which genes regulated by TNNI3K could influence the mononuclear percentage? TNNI3K is found on chromosome 3. We were looking at SOX2 as a possible candidate, and it's normally on chromosome 10, but there is some trans-EQTL located on chromosome 3 near the TNNI3K locus. This led us to our first experimental design, where we wanted to compare mice with high versus low mononuclear frequency. To do this, we screened the candidate, which was SOX2, isolated the samples, and conducted qPCR analysis to check the relative gene expression. Now, as presented before, there are 120 different strains of mice, all with different percent mononuclear cardiomyocytes. So we decided to choose Black 6 as our control. They have 5% mononuclear cardiomyocytes, which is pretty low. and our experimental was SWR with about 14% mononuclear, which is pretty high. And we isolated the adult cardiomyocytes from the heart's left ventricle. Next was conducting the qPCR analysis of the candidates. As we would expect, our results showed that TNNI3K had an inverse relationship with the mononuclear cardiomyocyte percentage. So as you can see, black 6 mice, which have low mononuclear cardiomyocyte percentage, had a much higher relative expression of TNNI3K. This is just what the paper showed us. Next, we looked at our candidate, SOX2, and interestingly enough, it showed the same relationship TNNI3K has, with a black 6, low mononuclear percentage, having a higher relative expression of this gene. This led us to our next question. Does the knockdown of TNNI3K affect SOX2? Is it possible that TNNI3K is an upstream regulator of SOX2? To do this and answer this question, we created an siRNA knockdown experiment. siRNA, or small interfering RNA, leads to temporary protein synthesis inhibition, so TNNI3K will not be expressed. First, we plated neonatal rat ventricular myocyte cells. We added the siRNA for TNNI3K, so the relative expression will decrease, inhibiting the protein synthesis, and then conducted qPCR analysis. Now, as you would expect, since the siRNA is inhibiting the TNNI3K protein synthesis, the relative expression of TNNI3K should decrease, which is exactly what we saw in the knockdown model when we did the qPCR. Next, we looked at the SOX2 relative expression, and we saw that this decreased as well. So the knockdown efficiency of TNNI3K was 60%, and the resulting SOX2 decrease was 50%. This shows that TNNI3K is likely affecting SOX2 expression. Now, this is an in vitro model. The next step was to take it in vivo, in mice. What we did was we obtained a mouse with TNNI3K completely knocked out, 0 relative expression. And then we conducted qPCR analysis once again. Now with a complete knockout model, 0% relative expression of TNNI3K is expected, which is exactly what we saw from the qPCR analysis. Then we looked at SOX2 relative expression. And interestingly enough, this decreased as well. The SOX2 decrease as a result of the TNNI3K knockout was 50%, just like our knockdown. So this confirmed the results from the in vitro, now in vivo. This takes us to the future directions. We saw throughout this experiment the past 2 quarters, the TNNI3K is affecting SOX2. The next question is how? Through what mechanism? What are the protein-protein interactions? Leading us to answer our big question, hopefully, how does this affect heart regeneration? I would like to thank the Lucis Lab, my faculty mentor, Jake Lucis, and my graduate student mentor, Michael Judah. Thank you all for your time and attention, and I hope you guys enjoyed my research presentation.